I have an announcement. As you are seeing on the screen that there is a mini marathon day after tomorrow morning. So this is organized by HBNI. So I request all the participants to please consider taking part in it. This is mandatory on part of HBNI. Okay, thank you.
Good evening, everyone. Welcome back to the seventh session of this theme meeting. In this session, we'll have a lecture for which I would like to invite on stage Professor H.S. Subramania, Director, Institute of Bioinformatics and Applied Biotechnology, Bangalore, and Biocon Chair. However, before I invite him to the podium, I request Dr. Majumdar sir to kindly introduce him to the audience.
Good evening, everybody. Good evening, Professor Subramaniam. Welcome to RRCAT. You know that Professor Subramaniam, when I approached, he was dilly-dallying a bit. But when I talked about HBNI and the importance of theme meeting, so then he readily agreed. And he has come all the way at a very short notice. He has come all the way from Bangalore to Indore, and today there is heavy rain in Bangalore. And in the morning, he was not certain whether he will be able to make it. But ultimately, it is his willpower that has made it possible. Okay, now Professor Subramaniam is as already Nidhi told, that he is the director of uh, this Institute of Bioinformatics and Applied Biotechnology, Bengaluru. Now, let me say a few words about his scholarly eminence. Professor Hosahalli S. Subramania obtained his PhD in 1993 from Molecular Biophysics Unit of Indian Institute of Science, Bangalore, in the area of structural biology. His doctoral work was on determination of three-dimensional crystal structure of a plant virus. He moved to the Oxford University, UK during 1993 and completed two postdoctoral studies. The first study was at the Laboratory of Molecular Biophysics, that is from 1993 to 1995, and the second study was at Sir William Dunn School of Pathology, that is from 1995 to 1998. During this period, his research was mainly focused on the molecular understanding of DNA replication pathway. He worked on some of the key enzymes in this pathway, including DNA ligase, DNA helicase, and DNA recombinases. The results of these studies were published in prestigious journals, including Nature and Cell. He joined the Central Drug Research Institute in 1998 as assistant director, and there he established the structural biology facility. His research interest at Central Drug Research Institute was mainly on the proteins critical for the survival of Mycobacterium tuberculosis. He, along with his group, worked on several important proteins of Mycobacterium tuberculosis and determined the structure of a novel enzyme from Mycobacterium tuberculosis. He joined Origin Discovery Technologies Limited in 2002 as in charge of structural biology. Since then, he has held various responsibilities at Origin and was senior vice president responsible for drug discovery and development before leaving Origin in 2014. During his career at Origin, he has led several drug discovery projects in various disease areas such as cancer, inflammation, infectious diseases, and discovered novel lead candidates. Currently, Professor Subramania is working as the director of Institute of Bioinformatics and Applied Biotechnology, that is IBAB Bangalore. He has over 50 publications in peer-reviewed reputed journals and is a co-inventor in 19 international patents. So with this brief introduction, I request Professor Subramania please to come onto the podium and deliver his evening lecture.
First of all, let me thank the director of RRCAT for inviting me for this HBNI meet, and several colleagues who have been constantly in touch with me and encouraged me to attend this event, Dr. Majumdar, Dr. Tapas Ganguly, and so on and so forth. Of course, thanks for all the colleagues who are assembled here, students um, who have come here from various constituent organizations of HBNI. Although I don't really deserve all the things that Professor Majumdar said, I humbly accept. Thank you very much for the kind introduction. So what I'm going to talk today is not really so much from the perspective of science or technology. I just wanted to give a glimpse of how do we really take up a project and how do we really execute a project within an industry. We focus very differently as compared to academic organizations. We ask very specific questions, starting from why this project has to be done, if it has to be done, how it should be done, and what is the real outcome, how much time it will take, how much money it will cost. Does it really make sense to develop this project at that kind of expenditure and timeline? So I talk more from the perspective of an industrial project rather than just from the science perspective of the project I'm going to talk. Before moving into the actual project, I just wanted to give a brief intro into the drug discovery sort of industry, the scenario of discovery of medicines. You know, everybody knows that it is really a very expensive business. More or less, it may cost about $1 billion to develop a drug. This is just a statistics from Forbes to show that how much it costed for some of the best companies in the world to develop the drugs. But it should not be really treated that developing one drug would really cost this much amount of money. That is not correct. What really is the cost really reflecting here is the cost of failures. Okay? For every 10 drugs which enter into clinical trial phase, 9 will fail. Of course, you have put in efforts, you have developed it to a certain stage, you have spent a lot of money, and that has failed. What is really shown here is about how much is the cost of failure rather than the cost of success. Okay? So that also sort of brings into discussion then how efficiently you need to run the project in order to really cut the costs of this failure. Launched in the US, it could, from an Indian perspective, it's more or less not affordable for most of the Indians, I would say. It would cost crores of rupees for the treatment. People do ask the question, like, would it cost so much to produce the drug? No. Cost of drug production is minimal, probably about 5% of the cost of what it is sold for. Next. Leave it. So in fact, what happens with most of the industrial projects is you don't really have the freedom to present it the way you want. Okay, you are limited with what I can present is certainly limited. I can't really tell some of the inside stories of how it was done, or I can't divulge the technical details of how it was carried out. But nevertheless, I will point to certain questions that we ask and how do we progress addressing those questions. So this project is all about discovery of a novel and potent Fabi is a protein or an enzyme inhibitor for treating multi-drug resistant Staphylococcus aureus infections. So obviously everybody knows that bacterial resistance is a serious global threat to healthcare. There is a lot of morbidity and mortality in community and hospital settings. Multi-drug resistant pathogens such as methicillin resistant Staphylococcus aureus or vancomycin resistant SA or methicillin resistant epidermidis, they're all major concerns. There's a high prevalence and virulence. There is a lot of bloodstream infection, soft tissue and skin infection. In US alone, there is about 20,000 deaths which are happening due to MRSA, which is not treatable because of drug resistance. Clinicians have very limited therapeutic options. The lack of interest by big pharma in antibacterial drug discovery because it's not really a long-term treatment. Pharma will be very interested in developing a drug if it has to be a lifelong treatment, like hypertension or diabetes. You keep taking it throughout your life. If you can treat it within 4 or 5 days, obviously there is very limited profit to make. So it's therefore, see, this is where we start the industrial project to see what is the relevance of the project. So obviously there is an unmet medical need and there is a lot of deaths which are happening. Particularly it's happening in the US. Therefore, there is money to make. This is how the pharma companies would think. So there is therefore a compelling need to develop drugs acting via a novel mechanism of action to combat the disease. Novel mechanism is needed because there is already resistance. If you develop another Me Too drug like methicillin or something like that, it will also have the same resistance issue. Therefore, you will have to tackle a new pathway which is not treated before such that there is no resistance developed against that pathway till now. So this Fabi enzyme belongs to fatty acid synthase pathway. This is another reason why we choose. When we choose, we start thinking about, because it is a drug, what is it differentiating between the pathogenic enzyme versus the host enzyme. If humans also have the same enzyme, if you inhibit the pathogenic enzyme, you are going to inhibit the human enzyme also. That means you have a potential for a risk, safety risk. Therefore, we try to evaluate the drug target. What is its potential to cause toxicity? Not just about, it's important. How is it different compared to the host enzyme? So this is the answer to that. The Fas2 pathway is actually relatively unexplored. You can see some 2 or 3 drugs which are already present, but most of the other enzymes in the pathway are untouched. These fatty acids are anyway essential for the bacterial growth. The components of Fas2 pathway are highly conserved, and reactions are performed by individual enzymes in bacteria, whereas in humans, it is a single multi-complex enzyme, very different than the Fas2 pathway in bacteria. Therefore, the chances of causing toxicity are extremely limited because they are completely different enzyme systems. The humans will use Fas1 system, which is a multi-complex enzyme, whereas bacteria uses individual enzymes for each step, which is very different as compared to Fas1 system. So the last reaction here is the elongation, which involves the reduction of enoyl ACP by this enzyme, enoyl ACP reductase, which is named after the gene. Fabi is the gene, fatty acid biosynthesis gene, I. So clinical success has validated Fab-I as one of the most attractive targets of the FAST2 pathway. That's the reason why we started. I won't get into too much of biochemistry of the enzyme. This is an NADH-dependent essential enzyme, catalyzes, as I said, reduction of 2-enoyl acyl carrier protein, ACP, belongs to the member of short-chain alcohol dehydrogenase or reductase. There is no alternative enzyme or rescue pathway available in Staphylococcus. There is only one enzyme. If you knock it down, there is no survival of the Staphylococcus aureus. The overall structural homology between Fab-I is high. There are conserved across gram-positive and negative species. And since the crystal structure is available, we can employ structure-guided design for development of the compound. So this is where we start with the objective is to identify a novel Fab-I inhibitor. We can design novel analogs based on reported inhibitors. We can use structure-guided approach, synthesis of pool compounds to validate biological assays, and then we'll have to be very worried about, you can develop it, but is it patentable? That's the question you'll have to address. So in an industrial project, from the day one, we start doing the patent search. We cannot develop anything without being sure that you have an intellectual property that can be protected in the end of the project. Otherwise, there is no scope for doing the project. And design novel chemical series like this. So these are the three known compounds at that point in time. There is an aphenium compound, which is very potent, in fact. And there is a crystal genomics compound, which is not as potent, but still potent. And there is another compound like this. The three compounds were available at the beginning of the project. Now the industry will ask the question, okay, since all these three compounds are there, what are you going to do special about it? What are you going to develop? It's already there. There are three compounds and there is nothing interesting about it. So if you look at really the properties of these compounds, look at this property particularly. Okay? So you have very low solubility for all these three compounds. Look at this line. You have to really dose them very high. Okay? This is because of two reasons. Because of poor solubility, there is very poor oral absorption. Because there is very poor oral absorption, you need to give very high doses of the compound to achieve sufficient concentration in circulation. So we talked to a lot of clinicians about this. So once we thought that is an interesting project, next thing we do is we will call a meeting of stakeholders. Who are our stakeholders? This is actually hospitals and doctors. So we had a meeting with hospitals and doctors. What they really identified is when you develop a new or novel antibiotic compound, you don't really use it for day-to-day infections. Day-to-day infections, you will give a broad spectrum antibiotic and you treat it. Usually an oral tablet like maybe amoxicillin, maybe azithromycin, maybe some other thing, right? Novel compounds are used in hospital settings when you cannot treat the disease with regular antibiotics, you use novel antibiotics, right? So what doctors were saying is, see, once you are in the hospital, you prefer an IV route of administration as compared to oral route of administration because your health condition is so poor, probably you won't be able to swallow a tablet. You will vomit or you are unconscious. Okay? You are already into a serious phase of the disease. Therefore, clinicians prefer an intravenous administration of the antibiotic rather than an oral administration of the antibiotic when you are in an ICU kind of a setting. That's where you are trying to treat. So that means to say that if you have to really have an intravenous administration, compound will have to be very soluble. Unless the compound is soluble in a saline kind of a system, you won't be able to do an intravenous administration. Okay? So this was our major differentiator. Okay? So that's the original question I said. Why are we doing this project? Is because these compounds have no potential to become an intravenous administration. These compounds can only be oral, it cannot be intravenous. Therefore, we have a scope. If you develop a compound which is very soluble, you can position it for intravenous administration. None of these compounds will do. Automatically, it will also address this issue because now it is soluble, therefore more orally available, and therefore you need lesser amount of doses as compared to this compound. So these were the two fundamental bases for the beginning of this project. This is what I said, how the pharma thinks. You will have to have a very clear objective. Why are you doing this project? We are doing this project because we wanted to develop an intravenously administratable antibiotic, and we want to reduce the dose of administration of these compounds. So these were the objectives of this project. So we could really determine the crystal structure of one of the compounds bound to E. coli enzyme, Fab enzyme. We could really map all the interactions that this compound is making with the protein. As you can see, there are two major interactions. One, there is a hydrophobic cavity here, and there is one solvent exposed cavity there. And therefore, we had very clear chemical design plan as to how we can really achieve our objective of having higher solubility and stability and oral bioavailability of the compound. These were our objectives, and therefore, from the crystal structure, we could really find out, okay, where are the regions that can be modified in the way of achieving the objective. That means we will be able to modify in this region where there is a hydrophobic pocket, which will enhance the activity of the compound. And this is in particular the region where we can modify to introduce higher level of solubility. And possibly, I will come to this, this is the region which is responsible for instability of the compound. So this is how we started off. What is most important here is this region. There is an unsaturated double bond there. Okay? And since this compound is, not really rigid with an unsaturated double bond and a carbonyl group there, we predicted that this compound is not going to be stable in the biological system. That means your liver is going to metabolize this compound because you have the presence of this unsaturated double bond, and adjacent to that, a highly electronegative oxygen atom. So this is a recipe for metabolism by liver. So we predicted that this compound is going to be highly unstable. So one way of stabilizing this compound is now introduce rigidity by just cyclizing between here and here. Okay? If you make it a cyclic compounds are always far more stable and less degradable. So that's exactly what we did. You can see that all that we did is we just cyclized that region of the compound into an azitidine ring rather than having an open ring with nitrogen open here like this. So using nitrogen, we just cyclized that series. Okay? And then we systematically analyze what happens if I change this group into various groups, and this side if I change this group into various groups. As you can see, what happens is really, if you don't have anything in the hydrophobic pocket, all the activity is lost. There is no activity. The moment you introduce the hydrophobic group, which occupies the cavity, you can see that the activity comes back. Okay? These are the numbers IC50 in terms of biochemical activity. These are cell-based activity. That means bacterial MIC assays. Okay? So you can see that the activity comes back. So again, we determined many other crystal structures, modeled several of them using computational biology approaches to see what happens. You can see that without a hydrophobic cavity, the compound ends here, and there is nothing which is occupying this region. That is the reason why you lose the activity if you do not have anything to anchor in the hydrophobic region. So we systematically, as I said, modified with several groups. As you can see, the potency increases with the presence of hydrophobic groups. If you have a fluoro group adjacent to that, the activity increases. You can see that now it is having a sub-micromolar cell-based activity. Similarly, methyl group also gives certain activity. And we also tried many other biaryl compounds. You can see all these compounds have various types of activities. And most potent compound, as you can see, is this one. When you have a benzofuryl kind of group, with a methyl group attached in the ortho position, you can see that this activity is extremely good. Of course, we don't know yet whether our objectives are achieved. Our objective is not really to develop a potent compound at all. Our objective is to develop a very soluble, very stable compound, which we can position it for IV administration. That was the objective. This doesn't tell about that. Whether it is really useful or not, I will come to how we assess that. So then we determined more crystal structures and again modified this solvent exposed region. As you can see that, we do have modified this region and we developed many compounds, by modifying that region. And again, these compounds appear to be very, very potent. I won't get into the structure activity relationship here. This is what is an interesting slide. Okay. Look at this activity. What this activity is actually telling you is the stability of the compound in a liver microsome. Liver microsomes are the ones which produces cytochrome P450 enzymes, which will metabolize any drug compound. Every drug is metabolized by some cytochrome P450 or the other. Okay? So we did this study using, the liver microsomes to see what happens to the compound if you incubate the compound in the liver microsome, which are producing cytochrome P450s, which are responsible for metabolizing the drug. This is the competitor compound, as I said, from the Aphenium Pharma, which was very potent. You can see that by about 60 minutes, hardly any compound is left. Compound is degraded by about 60 minutes. In contrast, see our compounds. This compound is 83% stable, even at 60 minutes. This compound is over 80%. Many compounds are much, much better, except this compound, which was poorer. But rest of the compounds, when you cyclize this central group, appear to stabilize the compound. That means they are not really metabolized by cytochrome P450s anymore. That means the metabolic stability of the compounds which we develop are far superior than the competitor compound. That's the objective number one. Objective number one is to enhance the metabolic stability, and therefore we have to dose lesser. So therefore, we have achieved the objective since our compounds appear to be far more stable than the competitor compound. This is the pharmacokinetic profile. So of course, liver microsome is one thing. When you inject it into the body, is some other thing. It behaves very differently, right? So these are the numbers. AUC is actually the area under the curve. That means how much of drug is in circulation is measured by area under the curve. The maximum concentration achieved by the drug is Cmax. This is the half-life of the compound within the biological system, and so on and so forth. Again, this is the competitor compound. Now compare with respect to this. This has a 2 microgram per mil area under the curve. We have almost 50 times higher amount of drug available in the circulation as compared to this compound. Similarly, the maximum concentration is 30 times higher, and half-life is definitely increased at least 2 or 3 times. So our compound behaved in the manner that we wanted in the in vivo system in the mouse models. So therefore, our compounds appear to have achieved the second objective. These are the ones which will decide the dosage. Therefore, I'm pretty sure that our compound would not require more than 10 milligrams as compared to 1000 milligrams that is administered by this compound, because it has a very low AUC, it has very low maximum concentration and lower half-life. As a consequence, you will have to dose it much higher. Therefore, we don't have to really dose it that higher. So then we optimized the compound. I won't get into that. And we did various modifications and finally developed a clinical candidate. I can't show the structure, therefore I have put XXX. And this compound has excellent properties, as you can see. It has the metabolic stability of 100% at 1 hour, which we wanted. This is what is very important. You had seen earlier, the compound is not even soluble up to 10 micrograms per mil concentration. Now you see this compound. This compound is about 20 times more soluble than the original compound. Therefore, we have achieved our objective in terms of being able to use the intravenous route of administration. Excellent pharmacokinetic properties, excellent bioavailability. As a consequence, the dosage that we needed was only about 0.9 milligrams as compared to, 1000 milligrams of the competitor compound. So we tested in the animal models. It's one thing is to say, another is to show it. You can see that our compound, this is our compound, okay, as compared to the standard of care linezolid. This is an existing antibiotic which is used for treating the Staphylococcus aureus infection. You can see that, we have excellent activity in the intravenous route at about 0.7 milligrams per kilogram, whereas this compound would require at least 4 times higher to achieve the same result. Okay? What it is really showing is the efficacy of the compound in the in vivo model. Our compound is far more efficacious compared to existing antibiotics. We also tested in the lung infection model. Again, you can see that our compound is far more efficient in reducing the bacterial counts in the lung. This is the log reduction in the bacterial count. You can see that, our compound really reduces by 4 log times, which is, 10,000 times reduction very quickly in about 24 hours. That's what we did. So in 24 hours, you are able to reduce the bacterial count by about 10,000 times in the lung. As compared to that, the standard is much, much poorer as compared to our compound. So in summary, this is what it is. We designed a novel and potent azitidine based inhibitors. We use structure-guided approaches. Again, some of these approaches, if you want to really use efficiently, you have to have an appropriate technology platform. We must have solved about 50 structures in this project. We did everything in synchrotron facility. Of course, at that time, we did not have access to synchrotrons in the country, so we had to really ship it outside of the country to collect the data. Unless you have the ability to really get the data very quickly, these projects will lose meaning. You can't do it next year. You will have to do it now. Either you do it now or you give up. So those are the two options. I think having facilities or technology platforms would really help in terms of moving projects like this, which are highly competitive, and you will have to do it with a given time frame. You can't just wait for next year to do this project. And, the compounds had excellent property, and now this is currently under clinical development. It's going through the clinical trials. Hopefully, one day it will come out as a drug. So that's the dream of any scientist who is working in the drug discovery space. But having said all these things, I just wanted to draw some more attention to one or two other points before I conclude, is that despite this kind of focused approach, that means starting from the beginning, you have a focused project, you have very well-defined objectives, and you have timelines, and you are using, modern techniques to, or technology to move the project. Still, drug discovery is not a simple business. Still, you can't really predict whether a compound is going to be efficacious or not. Still, you can't say that, a drug is safe or not, even after it is launched into the market. There are so many examples that a toxicity related to a drug will be discovered several years later after its launch. So you can't do anything about it, right? So now there has to be still a need for a paradigm shift in the drug discovery industry. But now there is a next phase of technological developments which is entering into the field, is through the data. There is a lot of data being generated, be it from the genomics data or a lot of clinical data such as imaging data or various medical diagnostics data and so on and so forth. A huge or a vast amount of data is available in the public domain. In the same project, what I said, even now, for example, when we dock a certain compound into a protein structure, how would you evaluate whether it is going to be experimentally valid or not? You'll have to still do an experiment. Yeah, of course, you can use the docking, which is a computational method, maybe only takes half an hour, 1 hour to do this. But what is the guarantee it works? There is absolutely no guarantee it works, right? So now the paradigm shift that is happening is, can we utilize some of the modern algorithms which are coming in terms of artificial intelligence or machine learning, to really extract more information from the biological data that's available out there and apply it into the drug industry. For example, if you want to really validate the target, there is a lot of genomics data available. Can you really analyze the genomic data and say whether a particular protein you are using as a drug target can be efficacious or not? There is a lot of modeling that is available to model the pathways, enzyme involved in the pathway, and see whether that protein is really meaningful. Can we apply the artificial intelligence and pattern recognition to say which of the compounds which are docked may be more meaningful as compared to others? Can we predict the toxicity? Can we predict the safety of the compound? And so on and so forth. So this is the current area that we are focusing at our institute, to develop algorithms and tools which will utilize a vast amount of public data that's available to extract meaningful information that may be useful for drug discovery research. Okay? With those words, I would like to conclude today's talk. Again, I'm really thankful for all of you. Thank you very much for inviting and listening very patiently. Although scientifically it may not be really very insightful or whatever, very interesting, but certainly it shows you an example of how pharma thinks, or how an industry thinks, how we ask the questions from the beginning about the validity of the project or the objective of the project and the timeline of the project, and try to achieve it in a timely fashion. These are the people who are involved in the project. And finally, thank you all.
Sorry. Professor Subramania is ready to take some questions. Okay. Yeah, please. Just a moment.
Thank you. It was a very nice run through of a successful story. When you mentioned about the pathway analysis to help you decide whether the drug can be either the toxicity can be reduced and whether it will be efficacious using pathways, do you mean to say that you want to look at cross-reacting targets? Because yes, the data will be useful in identifying whether the target is valid in a substantial population, if that's what you mean by that. But how will you know whether it is efficacious or safe by using the data?
So there are two approaches we are using. There is a lot of compound related data available, because as I said, 90% of the drugs will anyway fail, right? So there is a lot of failed compound and the data related to the failed compound, right? So can we now assign or attribute certain features to toxicity within a compound? Existence of a certain group or existence of a certain atom or whatever, okay? So we can really, one way of doing it is to look from the compound perspective, data available about a large number of chemical structures, because so many have failed, and we know that these are all failed due to various reasons, and can we really recognize a pattern of failure, why the compound failed? The second approach that we are doing is build the sort of network models of proteins and pathways. Okay? So if the compound has failed, it can fail because of two major reasons. One is it can fail because it interacts unintendedly with something else. Okay? It's not specific. No? That's why there is toxicity. The second reason why it can be toxic is it is specific, but the pathway you have chosen, by inhibiting it, you will invariably get into toxicity. No matter what you do, it is a mechanism-based toxicity rather than anything else, right? So the mechanism-based toxicity we try to address through the network and pathways. We construct a lot of networks and pathways of proteins, how they interact. If you modulate the activity of any component, how the other nodes are really impacted. So that's the other way of looking at toxicity.
Thank you. I'll just have one more question. So I couldn't help notice that in your structure determination of the compounds that you engineered over the time, one of them with the extra ring structure in the hydrophobic region, it came out of the pocket rather than interacting with the aromatic residues. In your final compound, did you go back to the original single ring structure or the double ring structure was retained?
So what we tend to do is we tend to develop this so-called structure-activity relationship, right? Of course, the first set of data is very preliminary. We have only few data points to really train this set. Okay? So what we try to do is each time we determine the structure, we go back and compare it with our hypothesis, right? So we thought that by adding a group here, the activity would increase, but it didn't. We solve the structure and see why it did not increase, right? And train the set now with the new hypothesis. Hopefully, that's more powerful than the previous hypothesis. That's all.
Thank you.
Yeah. See, it was a very nice lecture. See, I have just you changed the. You said that the alpha beta unsaturated amide is actually getting sensitive to biological
The
degradation.
alpha beta unsaturated amides are typically prone for metabolic degradation.
Yes, but when you are putting this agitated as amide, it is the stability
But
is
because it is cyclized now, the nitrogen is no more a part of the linear chain. Okay? This is not now connected to the alpha beta unsaturated amide. It is rather a cyclic amide.
And then suppose if you selected a four-member amide, four-member amine amide, if you reduce the size to three-member or five-member,
We tried everything.
Okay.
We starting from 3 to, we went all the way up to 7 or 8. Okay. So
four-member.
there what happens is the space availability, right? As you increase the number of carbons, the size of the cyclic group goes up, right? And there is only certain level of tolerability on the size. Okay? We did try starting from 3 to 8. 3 works. 4 is probably the best. 5 is almost equal to 4. Above 5, it doesn't happen. You start losing the activity because it doesn't fit. There is not enough space there.
And also you said that you are increasing the your aim was also to increase the solubility. So if you change that, now then maybe other parameters will also might change. So
I didn't get that
see,
question.
you said that you want to improve the solubility of the your drug molecule or whatever molecules you have developed.
Right.
But it might change the other properties of the.
Correct. So that's why the lead optimization is not a single parameter optimization, right? If you isolatedly look at it, we had many compounds which are even more soluble, right? There are some compounds which are even more stable, okay? But we can't really take that, right? We have to take a compound which is balanced, okay? Of course, there is give and take when you take about balance, right? We compromise a little bit on solubility, but we get much better metabolic stability, okay? We compromise a little bit on metabolic stability, therefore you get much better safety, okay? Like that, lead optimization always works as a balancing act between various physicochemical properties rather than one property being much better than anything else.
A very insightful lecture, actually. And I'm still thinking and considering some of the insights that you brought in right in the beginning regarding the cost and effectiveness of drugs. So the thought that I am coming through is like, like what you are saying, the bacteria antibiotics. So it's the profit margin for the companies is a question because of its short nature of the treatment versus cardiology or cancer medicines, which are for longer term, but they are still very costly. Uh,
Still?
Still very cost effective, still very costly affair for the patient perspective. You say the treatment cost is much higher for cardio or cancer treatment. So why it is so? Like even though the drug will be sold by the company for longer period of time, so why the price do not come down for those kind
The
of
price of our compounds is not decided. It's still in the clinical trials.
Yes.
I'm hoping that if it really becomes a drug, it will be very cheap as compared to anything else. Our compounds are not there in trials. As I said, the cost of a drug is not about the producing cost of that drug, okay? Or for example, if you take number of people worked on that project, how much money they spent, add all those together as a project cost. Okay? It's not about project cost. Okay? Project cost won't be that much. Okay? Even if 100 people work for a year, it won't cost $1 billion, right? That's not the thing. The problem really is cost of failure. Okay? When a company is working, company is not costing it based on successful examples. Okay? Company is basically on all the projects. If 90% of the project fail, okay, each project, let us say, spent $100 million, you lost $1 billion.
So meaning the fishing game is more in cardiac or cancer treatment.
So the answer to that should be that how do we reduce the cost of failure?
Yeah.
That means instead of 9 out of 10 drugs failing, can we make it only 3 out of 10 will fail? That's where I was saying that we are now trying to develop tools which are capable of predicting, okay, these are the compounds which may be more successful. Hopefully, even if you marginally reduce that cost of failure, the cost of drugs will come down.
Here. Okay. I guess you touched upon it a little bit towards answering the question just right before this. But when we talk about these multi-drug resistance, I mean the antibiotics against multi-drug resistant strains, I mean, we have to consider the patients also to whom these are administered, not just the delivery of, I mean, whether it is going through IVF or through oral pathways. Rather, that because a lot of these, patients who suffer from bilateral kidney failures, they during their course of prolonged dialysis, etc., they are suffering from these bacterial strains. So their efficacy of actually metabolizing and getting them out of the system would be much less. So the toxicity would be way higher than in a normal person like you or me. So how, I mean, network analysis, structural analysis, all these are fine. But how do you consider these also when designing such drugs?
I don't know whether I got your questions completely correctly or not. See, when we talk about toxicity, we will always talk relative to the disease area. Okay? The standards of a drug being approved is dependent on what area it is. Okay? For example, if you take the so-called orphan drugs, we call, means for a specific disease, there is no available drugs. And anyway, patient is going to die because there is no drugs. Yeah? The standards of safety for such kind of diseases is very low. people don't really care so much about it because anyway patient is going to die. There is no treatment available. Therefore, cut the standard of safety to some extent and give something, that's the thing. You take diabetes or hypertension, standards are somewhere in the moon. It is very hard to now produce an anti-diabetic or an anti-hypertensive because they are very treatable disease as of now, right? You need to have a far higher safety standards. So when we talk about the safety and toxicity, we always take it into account what disease you are trying to treat. Okay? Similarly, for antibacterial, it is not really as high again, basically because it is a very short-term treatment. Okay? Typical antibiotics are given only for 5 days. Okay? So, a little higher level of toxicity is allowed in the area of antibacterials. Okay?
So just going back to the question that where you stopped, and I think it was discussed earlier also. As you mentioned, there are two types of toxicity or undesired side effects that are possible. One is mechanism based because the target
I'm
that
unable to hear at all.
Are you able to hear me?
I have a little bit of a hearing problem. If you can come a little closer and tell, I'll be happy.
Yeah. Yeah. Can you hear me?
Okay.
Yeah. So basically, in case of mechanism-based toxicity, this is, as you said, because the target that you have is inherently, expected to interact with some of the other pathways that may lead to toxicity. On the other hand, the other aspect that you mentioned, that where you are trying to connect an atom or a functional group or some pharmacophore kind of thing to other possible pathways where this might be leading to, and then leading to failure. So for that, we, as I understand, we mostly have in typical this drug development process that the structure of this molecule and then you do some of the
No.
typical assays.
So what we do is we again try to use a lot of publicly available data, right? There's a lot of differential gene expression data available, right? In a specific disease condition, which are the proteins which are impacted. In fact, nowadays you also get a lot of data, or we generate data ourselves. The changes in proteomics or the gene expression levels in response to treatment of a compound. Okay? We usually do it in cell-based systems. We take the compound, treat the cells with the compound, and try to see what are all the changes that happen, right? So then, once we have sufficient amount of data, okay, we start sort of training the set in terms of identifying features versus modulation. Okay? That's why we generate the models, right? So in a particular disease, we construct the model. These are the proteins seems to be involved or at least modulated, right? They are either up or low, or up-regulated or down-regulated in terms of expression. Then we try to develop the data on compounds. When you treat the compounds, what is going to happen? We try to link these two together into a model. Okay? That's how we predict the toxicity.
Yeah, so that means you are generating data for all these candidates.
Yeah, we generate data to some extent on our own.
Okay.
Wherever public data is available, we use it.
Thank you. Thank you for giving the perspective from industry's inside thing, right? So my question is actually basically comment. So like, the cost of failure is related to that part. So, you are right that when you develop something with a very defined objective, correct? And for example, you have decided to develop a compound against the bacterial infection. And let's say it does not succeed, right? So my point here, why industry is little reluctant in India to collaborate with academia. So one compound which is focused for probably bacterial infection can work for other model system. So, academia had a lot of model system, cancers, especially I work in the cancer field. So if we check that there and, come out with a logical mechanism of action of that compound, how it is working to treat the cancer. So that can fail in one particular situation, but that it can be a success for other situation. But like in US, there is a lot of industry academia collaboration turn into a very successful outcome in the clinical development. Why in India we are lagging behind? Is there any?
You mean
I mean,
in terms of why the industry academia collaborations are not really developing in a way,
correct, correct.
right? That's what you are trying to
Yeah.
ask. Well, there is no one single answer I can really think of, in the sense that while I was in industry, I could see a certain resistance within the industry. Although we try to push the things together, the major thing is possibly industry works in very sort of fixed time frames and things like that, which industry has a feeling that academia won't really get abide by that kind of timeline. You are free thinking people, and for you, whether it's this year or next year may not make too much of difference, whereas industry will count every day and every hour and every minute. So that's probably one of the reasons why it doesn't work that way. The second, I feel after, and I worked both in academia and industry for substantial number of years, so I know a little bit more about what is really happening. I think academics are very good in terms of free thinking, maybe creativity and things like that, but they are very poor business people. You'll have to position it appropriately. It's not enough if you discover. You may have discovered the gold mine, but if you don't really project it as a gold mine, you're not going to get business, right? That's another problem I'm seeing with academia is they just don't know how to position it. You have to position it very appropriately. As I was saying here, let us say academia had discovered this compound. Nobody will take that compound, right? You have to say that, look, this compound, you can position it for intravenous administration. That's positioning. It's not about compound and its properties, right? It is about telling that this compound is very good for intravenous administration. That's the positioning. Compound is same, data is same. Positioning can be that, okay, here is a very potent antimicrobial compound. Here is an antibiotic which you can use for intravenous administration. That's where the difference is. I think it's very good if academia also, probably it's happening to a large extent in the West. For example, if you take the lot of startup clusters around MIT, Harvard, they are all academic scientists. They have learned the art of positioning it. I think in India, we also need to learn the art of positioning, selling.
Yeah, I understand. I completely agree what you are saying, that delay which is happening in the academic institutes because of the a lot of things, right, bureaucratic things. But one more thing I would like to ask, the industry status today in India. So industry does not want to take a lot of risk also. For example, taking a compound to clinical trial, and if it fails over there, is a zero. Everything is becomes zero, right? So that has, I don't know how much our industry, Indian industry, is ready to take that sort of risk to enter into the clinical trials rather than selling the compound to a bigger company.
Right. So I think India certainly started off with the so-called generic capital, right? So produce drugs which have off patent and then sell it much cheaper because we have a good chemistry manufacturing base. But that sort of concept is slowly changing, although I think that it is still not completely changed. But I can see many startup companies, particularly, we also incubate a lot of companies in our campus, and as well as I also interact with a lot of incubation centers. Therefore, I know which are the companies working on what. I can see that the trend, at least in these startup companies, is definitely moving towards developing their own, compounds and getting into clinical trial. But having said this, we don't have to really be too much worried about we have to do clinical trial for everything ourselves. It's not necessary, right? It depends upon how do you really make the business agreement. For example, when we also did a commercial deal with this particular compound. The deal we did is, okay, you take the US and Europe market, but Indian market remains with me. They cannot sell this compound in India. So I am the owner of the compound in India. Therefore, I can sell it for whatever price I want, right? So like that, there are many different ways of doing business deals, which is suitable for whatever your objective is. If you want to make a lot of money, there is one way of doing a deal. If you want to preserve your country and pricing in your country, there is another way of doing it. So we can do in many different ways. So just because we don't do clinical trials ourselves doesn't mean that the ownership of what you discovered is less meaningful. It all context dependent.
Thank you.
Okay.
Thank you so much.
Yeah, sir, thank you very much for enlightening us on a very important topic of drug development. Hello, am I audible? Yeah. First, I would like to thank you for enlightening us on the process of drug development, which is definitely there is a lacuna in India, as right now mentioned. Uh, right now, I have two short questions. One is that there are certain, when we are talking about the tools to predict the toxicity or the adiabatic property of any compound. So, what do you think? How reliable these tools are? There are some online servers are available. You put the chemical structure, they give you certain predict certain properties. So, first question is that how reliable these online softwares, and second is that because I am interested in this serum protein binding, we also work on serum protein binding. So I can see that the parent compound and the modified compound which have it has only slight difference in serum protein binding. So what do you think? How does this binding is important for the bioavailability of the compound?
Hello. Ah. So coming to the first question, these are sort of very nascent stages of you know development of these tools and things like that. I don't believe that it still has a very high degree of accuracy in terms of prediction as of now, right? But what we believe is as we have more and more data, yeah, it becomes more and more powerful. It just depends upon how much data is available, how much you are using, right? So to me, it is more of, okay, we are developing an algorithm. Hopefully one day it will have sufficient reliability to go ahead. As of now, the answer is, I'm not satisfied. No, I don't think it has really that level of reliability yet. Now, coming to your second question, what was it? Oh, about the serum protein binding. So while serum protein binding is one of the parameters, okay, ultimately what really matters is the ED50 calculations, right? How efficacious the compound is at what dose. Most of the compounds do bind to serum proteins, and in fact, if it doesn't bind to the serum proteins, it gets eliminated very quickly. So serum binding protein is a double-edged sword. Okay? If it binds too much, yeah, it will reduce the availability of the compound in the target site. Okay? If it doesn't bind, it will clear very quickly. Okay? So I believe it's the question of balance. Okay? There has to be serum protein binding, but it not be so excessive that it's not available at the target site. Okay? So there is no one specific number I can quote. Probably I would say that if it is about 2% available, that means 98% serum protein binding, that works.
Yeah. So I just have one query. This is more of a general kind. As you've been just mentioning through the question answers, you've been talking about the availability of algorithms, and you mentioned it in the last part of your talk, also availability of algorithms that can predict the character, the behavior of the compound, of the molecule that you're talking about. So does that also mean that this is essentially a replacement to the structure determination activities, or is it like there are two independent ways? That is, on one hand, you determine structures, and you determine structures only in a smaller subspace rather than the complete space. Is that the way you're talking about?
No, I think, see, There are two aspects here. Of course, if you have the ability to determine the structures quickly, it will move the project very fast. There's no doubt. Okay? As I said, we determined about 50 structures in this project. On an average, in my experience, developing this compound without these structures would probably take about 3 to 4 years to develop, because each time you need to synthesize a compound, go back to the biochemical assay or whatever assay, then again redesign and so on and so forth. Right? And you would require about 400 to 500 compounds to optimize to this level in a general project I'm talking about. Because of these 50 structures, we could cut it to half. We were able to optimize it in about 2.5 years, and we needed to only synthesize around 200 or so compounds. If you have an ability at that time, as I said, we were shipping it outside, it was not really efficient. Each structure would take at least 8-10 days to get it. Imagine that a platform is available where in one day you can determine all these structures. Yeah? It would definitely help in terms of moving the project faster. Ultimately, you may end up with the same compound, but in what time? That's where this structure helps.
Thank you for the wonderful talk on the industry perspective.
Sorry, I can't hear.
Can you hear me now?
Better.
Okay, so thank you for the industry perspective. I just had a question regarding the IP issues which industry faces. Is there more flexibility in the West on the IP issues when they engage with academia, which could be potentially a reason why there are much more academia industry collaborations? I'm just naive, I'm asking you.
No, when we work in an industry, we are working for only intellectual property, because that's the one which is sellable, right? You can't sell anything other than intellectual property. So companies will be extremely conscious about intellectual property, and they won't do anything. For example, each compound, when we design, before synthesis, I will have to show that there is patentability on that compound. Otherwise, you won't be allowed to synthesize the compound. Okay? So intellectual property is an integral component of every step. You will have to protect it. If there is no intellectual property, you will have to show that, okay, how are you going to introduce it in the next compound? That's the only way you will be able to work in an industry. Without intellectual property, no way.
So I have a small question. So do you have any small molecule library that the academic institutes can access?
Small molecule?
Library? Do you have any library that, like, Pfizer's or anything that the Sigma? Do you have any library that we can access, like as an academic institute, the collection of the small molecules? Like, do you curate the library of small molecules?
Do we have a library of small molecule? You're saying?
Yeah.
At of course, company did have, but I can't do much about the company's IP. But at IBAB, we have assembled about, let's say, 30 to 40,000 natural product compound library. Okay, and also we have assembled all the compounds available in the drug bank in a format which is suitable for docking. So you can use the 40,000 compounds in a virtual screening platform.
No, not virtual screening. I'm saying like real drug, like real molecules. Can we access them?
We don't have real molecules at IBAB. At company, yes, we had, but they won't give you the access.
Thank you.
Okay.
So I think you can discuss more during dinner. So thank you, Professor Subramania, for your wonderful talk and very erudite experience of industries and academia. And thank you very much. Now, just a moment.
Thank you, sir. Now, I would like to request Dr. S.V. Nake, sir, to kindly present a memento to Professor H.S. Subramanian. Thank you, sir. Here concludes the Day 2 of Life Science theme meeting. On the behalf of organizing committee, I would like to thank you for being with us diligently. I would like to thank all the dignitaries present here. Thank you. Let us all meet at dinner organized at the rooftop of the convention center. Thank you so much.